This video is Unit 8, 8.5 Gene Regulation. This is the last section of Unit 8, looking at gene regulation. What is that? Well, gene regulation has to do with gene expression. What is gene expression? It's when a gene expresses itself. We have learned in this unit that genes code for proteins, these processes like transcription and translation. You start with the DNA, you end up with a protein. That gene has expressed itself, right? Well, only a fraction of genes are expressing themselves at any one time. That makes sense. You can't have every protein being made all at the same time. And so those proteins that aren't being made are being regulated, which is the control of gene expression. Gene regulation is the control of gene expression. This can get pretty complicated, right? So we're going to keep it pretty simple for this class. We're going to keep things really simple. We're going to talk about how prokaryotes regulate gene expression, how eukaryotes then regulate gene expression, and we're going to add this cool little thing at the end because it's a cool little thing. Let's talk about prokaryotic gene regulation. Prokaryotes use a mechanism called an operon. The word operon comes from a portion of this operon. So what is an operon, first of all? It is a group of genes that are being regulated all at the same time. So maybe this group of genes has to do with a particular function in the cell that only needs to be used at certain portions or certain times. And so all of those genes are being regulated all at the same time. And they are all kind of turned on by this section called an operator, which you can see there. The reason that the operator is not able to work is because on the operator is something called a repressor. It represses it. It stops it from happening, right? And so usually there's some kind of trigger. One that is often used is the lac operon, which has to do with prokaryotes are breaking down lactose. I'm not going to ask you to know specific examples, but essentially, when is this operon going to work? When it comes in contact with lactose. When the cell comes in contact with lactose, that repressor is going to lift off the operator, and RNA polymerase is going to transcribe all those genes and make proteins. It's going to make those proteins. Those proteins are going to break down lactose. And when all the lactose is gone, that repressor is going to bind back to the operator and stop those genes from being expressed. It's a way to save resources. It's a way to save energy. It's just a way to make prokaryotes better than they were. What about eukaryotes? Well, eukaryotes are a lot more complicated, right? They get a nucleus, and so that makes things complicated, literally, because there's not as much. I mean, there has to be some stops in between, right? We talked about some of that stuff as we talked about transcription and translation. We're going to talk about things that make eukaryotic cells different, particularly lots more genes, lots more going on. Let's look at a couple of examples of this. So one of these things are called stem cells. A stem cell is an undifferentiated cell. All right, undifferent means same, right? And so you have these groups of cells that are all the same, meaning that they, at some point in their development, can become any kind of cell. Well, at some point in their development, they're going to do just that. A stem cell is going to become, say, a liver cell. Well, once it becomes a liver cell, it has differentiated, or another word would be specialized, right? And so once it has become specialized, it no longer wants to become a heart or like a finger or something along those lines, right? It still knows how to do all those things. It hasn't forgotten that information. The DNA doesn't become erased. Liver cells still have all the instructions on how to become an ear or a finger or a heart or whatever. They just don't do it. And so those genes are being regulated in that they're not being expressed. And the how to be a liver genes are being expressed, or at least some of them, right? They don't need to know how to make a liver anymore. They're already liver cells. They need to know how a liver functions. And so there's a whole lot of going on there, right? A whole lot of different functions, a whole lot of things going on. And so the big deal there is just, again, differentiation involves regulation. Hox genes, RNX. Hox is short for something called homeobox. You don't need to know that. If you're just wondering where Hox came from, that's where it comes from. And Hox genes are, the way I like to describe them, is they're like pattern and position genes. So what does that mean? Well, this is your typical vertebrate embryo that you see here on the screen. And a vertebrate embryo has a particular pattern to it. Why does it have that pattern? Why is its pattern not a leaf or a shrimp or, I don't know, something else, a worm? Because it's not, because these Hox genes have a particular pattern to them, right? And so then vertebrates are going to have similar Hox genes because they have similar patterning, but there's going to be some of those genes that are different. And you see that there are Hox A, B, C, and D, and so forth. There's going to be some differences, right? And so some of those genes are going to be expressed. Some of them are going to be regulated so that they're not being expressed. And then, of course, in other organisms, you just don't have those genes. And so that's what you're looking at in this particular picture. It's showing you some different versions, different organisms that can have different kinds of genes associated with that. And as you develop, these are going to change as well. This is a great example. Tissue repair is a great example. As you are developing, you're creating tissue. Great example would be like nerve tissue, right? But as you age, certain tissues are no longer repairable. Like if you damage your spinal cord, it is not repairable. Why? Because those cells don't know how to do that anymore. They're regulating those genes. And so that's one of the ideas behind stem cell research, is trying to figure out how to get cells to like remember how to repair themselves again, right? And so they still have that information. Those genes are just being regulated. And so if you can like unregulate them in some way, perhaps you could get them to express themselves again. You get the idea? And all of this has to do with cell signaling. Cell signaling is just cells communicating with one another. So let's say that a cell decides to be a liver cell. It's pretty important if you're like a stomach or a gallbladder cell, right? Because you need to know, that you need to be next to the liver. And how do they know what next to even is? It's not like they have eyes or ears or anything like that. They literally have to signal one another. And so cells communicating with one another are going to control gene expression, which is also going to control gene regulation. As they talk to one another, they're going to become certain things and not become other things. And this signaling is going to control expression and regulation. And then lastly is this concept of epigenetics. So anytime you see this think like epidermis. It's like outside or on. It's the surface of genetics, genes. And so these are things that can happen to the DNA that can control gene expression from outside the DNA. Rather than controlling the actual A's, T, C's, and G's, these are going to things that are like molecules or other kinds of chemicals that can be added to the DNA that control gene expression from the outside. There's a lot to do with epigenetics. A lot of studies being done about epigenetics right now, so it's a fairly complicated sort of thing. But the big idea here is that a gene can be expressed or regulated for another reason other than the fact that it codes for a particular protein, right? You can have blue eyes even though neither parent passed it to you, or you can have blue eyes even though brown eyes are dominant and one of your parents has both genes for brown eyes and that gene was being suppressed because of these epigenetics. There's a lot of examples of this. Big idea that I want you to take away is that there's some kind of change that can happen from outside the DNA itself.